Hi, everybody. My name is Marcelo Mira. I am a full professor at the Pontifical Catholic University of Paraná in Curitiba, Brazil. And the title of our study is Functional Analysis of Candidate Variants on Early Onset Leprosy using a Novel Cellular Model. So all started when we were discussing the possibility of using leprosy extreme phenotypes to further advance on the understanding of the molecular pathology of the disease. So based on that, we identified a family resident at the state of Piaui. It's in the northeast of the country, and displaying several cases of leprosy, including a pair of monozygotic twins diagnosed with leprosy at the age of 22 months. So very, very early onset. So we decided to study this family under the hypothesis that there is a strong genetic component controlling susceptibility to the leprosy phenotype in this pedigree. The study can be subdivided in three parts. On phase one, we did whole genome sequencing of the members of the pedigree, and we found 2 mutations on LRRK2 and 1 mutation on NOD2 that were the best candidates to explain that phenotype. Then on phase 2, our partners from McGill University, Erwin Schur's lab in Canada, did a functional analysis using a raw cell lines. And they found that there is actually a functional impact of the LRRK2 mutations on ROS production and apoptosis, and also a possible role for that NOD2 variant. And this work is done, and it's submitted for publication. And that brings us to the third phase of the study, which is to replicate these functional studies now using human primary cell lines. And this is what's ongoing and financed by LRI. So the idea is to use UPCs, urinary progenitor cells obtained from urine of the members of the pedigree, use these cells to reprogram them into iPSCs, induce the pluripotent stem cells, then edit these cells using CRISPR-Cas9, so we can create different settings, different combinations of the genotypes of the 3 variants that we are interested in. Then we get these cells and we transform these cells into macrophages. We stimulate these cells in vitro, and we measure a series of functional assays. More recently, we introduced a second branch to the study, which is based on the simple idea that we can identify individuals with the same genotypes of the Piaui family members. And then we can simply get blood from these people. We can make macrophages derived from monocytes, and we do the same functional assays. So then we don't need to do all the genome editing phase of the study. And we already identified a number of people with the same genotype as the father, as the twins, as the mother, and the grandmother. And we are actually working on this right now. So as for the study progress, in May 2021, mainly due to the pandemic, we requested a freeze to the LRI committee that was approved. Fortunately, in February 2022, we were able to resume the study with the pretty much the same goals. In general, we want to advance on the understanding of the role of the LRRK2 variants on the control of these immune in vitro phenotypes. But we also want to apply CRISPR-Cas9 to do that, associated with this investigation of cells of MDM derived from PBMCs from both the Piaui family members and leprosy-affected individuals and controls harboring the same LRRK2 genotypes. So this is just to show that even under a freeze, we were able in 2001 to produce some results. These are results of an experiment in which we measured the production of cytokines and also apoptosis by flow cytometry. This is apoptosis. This is production of a series of cytokines. We still have to do this again. Again, this is just to show that the pipeline of work is finally working. We also did a LRRK2 NOD2 co-localization experiment using cofocal. And we can actually see that if you look at cells from the father, who is heterozygous for one of the LRRK2 mutations and homozygous for the rare allele of the other mutation, and then you look at one of the twins who are homozygous for both LRRK2 mutations for the rare allele, you can and you compare them with a control that is homozygous wild type for the LRRK2 mutations, you can see that there is an effect, which is very, very interesting. We also managed to obtain UPC cultures from two members of the family, grandmother and one of the twins. These cells are already available in Curitiba and being processed to be turned into iPSCs and then genome editing and then macrophages for the functional assays. As an unexpected outcome, we managed also to obtain B lymphocytes that we immortalized, and we also have these cells available in Curitiba. And then in order to use these cells, we really have to sit down and think about the design of the experiments we want to do with these B cells. The good news is we do have this alternative model available. So this is the new time schedule in which you can see the pandemic colored in green, and then the period of the freeze and what we're planning to do from now on. So this has only been possible with the help of a lot of people. So at PUC, we have Alana, Maria Luisa, who are PhD candidates at my group. Irenice is helping a lot with the technical issues, and Dr. Leticia Fracaro is one of the co-supervisors working with us at PUC. Then we have the whole team of the Federal University of Piaui. Ana Lucia, Esther, and Semiramis helped a lot in dealing with the family, in getting the biological samples. We also have partners at the Institute, at the Carlos Chagas Institute, which is the Fiocruz branch in Curitiba. Patricia Shigunov, Rubens, and Isabel are helping a lot with the work with the UPCs. In Rio, Dr. Flavio Lara and Dr. Roberta Omo Pinheiro have been extremely helpful with the functional studies. Most of the results that you've seen here were done at Flavio's lab and Roberta's lab in Rio, Fiocruz Rio. And finally, McGill University, of course, Erwin Schur and his team, Monica Sauer and Young, they've done most of the all of the work that is published on phase 2 of the study. These are the institutions that I just mentioned, funding, of course, by LRI and the Turing Foundation. And then at the end, finally, we've got a few questions from the audience at the original presentation. The first question was, do you expect any differences in the cellular model in different settings? This is a very good question. Of course, that the genetic background underlying the effect of the LRRK2 mutations and the NOD2 mutations can have an impact on the functional consequences of these variants. That's why we are using two strategies. We are using the patients, and we'll compare individuals with or without the different combinations of LRRK2 and NOD2 variants. But we are also, and that's why it's so precious, the experiment with the Piaui family members and those cells, because then we want to do the genome editing. And by doing genome editing, we do have the same genetic background. We are only changing the presence or absence of the variants. So then we'll have a very clean picture of what the impact of these variants are. Now, is there also a control in the experiments? Of course, yes. When we use the general population of individuals affected or non-affected by leprosy, the controls will be in the individuals harboring the wild type genotypes for all the 3 variants, right? And when we are using the Piaui family members and we're doing the genome editing experiments with those cells, then the control is the unedited cell. And by doing the editing and having these different combinations of the genotypes of the three variants, we can do the comparison across the setting. And then finally, what was the stimulus in the cytokine experiment? We used MDP because we do have NOT2 there, and that proven to be the most efficient stimulus in vitro. So this is pretty much it. Thank you so much. And I hope I see you again next year. Bye.